Thank you. Can you see my slides and
Yes,
all
we
good?
can.
Great, thanks. Yeah, first I'd like to thank Julian for inviting me. And I don't know whether you guys know, but Julian and I were postdocs at around the same time at the National Institute for Medical Research in London, on the same floor of this grand old building, and we had some good times. And so that was more than 10 years ago. So yeah. Today I'd like to tell you about a story in the lab on non-autonomous regulation of stem cell proliferation, either in the developing fly central nervous system, and particularly how the glia niche, which surrounds these neurostem cells, can affect the ability of the stem cells to proliferate. Oops. So in general, my lab is very much interested in the effect of non-autonomous cues from the microenvironment and how this impacts on organ size control mediated by autonomous regulators such as metabolism, signaling, and transcriptional regulation. And we utilize the fantastic genetic model organism, the fruit fly Drosophila, to answer these questions. So what you're looking at here is the developing fruit fly Drosophila, and this is larval stages of development. We choose to study these questions in this stage of development because the larvae can undergo tremendous organism growth in response to dietary nutrients of approximately 200-fold from the beginning of larval stages to the end of larval stages. And what we are particularly interested in is how organs such as the central nervous system, the adipose tissue, and the muscles can cross-communicate with each other and coordinate the growth control of each of these organs in response to physiological changes, and also how these scale with the overall growth of the organism. But today I will be mostly talking about what happens within the central nervous system and how the local cues from the microenvironment that surrounds these neurostem cells can affect its ability to undergo proliferation. So what you're looking at here is a schematic of the CNS of the fly, and we're mostly working on these cells depicted in gray, and they are the stem cells or the progenitor cells of the Drosophila central nervous system. They're also called neuroblasts. And these cells are able to self-renew as well as differentiate to generate the neurons and glia cells that ultimately give rise to a functional adult central nervous system. So the story I'm going to tell you about today is spearheaded by this very talented PhD student in the lab, Joanna Dong. And in particular, she wanted to understand how the local environment of the neurostem cell can affect the stem cell's ability to produce neurons. And basically, as I've briefly mentioned, that stem cells are capable of self-renewing, this is mostly regulated by asymmetric cell division. And this is the control of asymmetric division is very much instrumental in maintaining an invariant number of neurostem cells that's maintaining the ultimate size of the brain. But on the other hand, the neurostem cells are also multipotent, and they're able to differentiate to give rise to two specialized cell types, neurons and glia cells. And the ability of neurostem cells, especially to produce neurons, is also instrumental to generating a functional brain. So these two mechanisms really go hand in hand to ensure that the correct size brain is produced at the end of neurogenesis. And one of the specialized cell types that's produced by the neurostem cells initially actually forms a microenvironment surrounding the neurostem cells. And as you can see from this movie, the stem cells are labeled in red and the glia cells are labeled in green. And these two cell types really intimately communicate with each other. In fact, the glia cells form these chambers that surround the neurostem cells. And it has been shown work from my previous lab as well as Andrea Brandt's lab about 10 years ago, that the stem cell niche really secrete signals that can affect the behavior of neural stem cells. And it has been mostly studied during early stages of development. And this glia niche is really instrumental in relaying the availability of nutrients by sensing, by relays through another organ, the adipose tissue of the fly, to relay the availability of nutrients to then, through the glia cells, to instruct the neural stem cells to only reactivate from a period of quiescence when nutrient is available. Therefore, this relay system really couples the overall physiology of the animal to coordinate neurogenesis only when nutrients is available. However, it is not really well understood what happens later on during later stages of neurogenesis. And Joanna, we really wanted to ask what happens in neural stem cell proliferation when you disrupt the stem cell niche. And she really takes the tact of asking, if you increase coarse glia overgrowth, what would happen to stem cells, and hoping that through this strategy, she would uncover some of these important cues. And so she utilized a well-defined model for glia overgrowth, and this is through FGF activation. And ectopic FGF signaling has been shown to be very much relevant in glioblastoma, where it causes not only the overgrowth of the glia cells, but also has dramatic effects on its neighbors. So therefore, what we're looking at is also relevant in a disease context. So what you can see here is if you activated FGF signaling in the glia cells, you've got a lot more glia cells in green. And so what she's hoping to find is she postulated by increasing the size of the glia cells, you would significantly increase the amount of signals that travels from the glia niche to neural stem cells and therefore affect the behavior of neural stem cells. So she looked at several parameters, including the timing of neuroblast cell cycle entry and exit, as well as asymmetric division. But the main phenotypes that she found is really the ability of neural stem cells to produce neurons. So she did two experiments to characterize this phenotype. And the first is through live cell imaging. And what she did here is she activated FGF signaling in the glia niche, utilizing a constitutive activated form of the FGF receptor called heartless in flies. And when she activated FGF signaling in the glia niche, she found that neuroblast cell cycle progression is significantly slowed down. So this is quantified here as the cell cycle length is significantly increased. And consistent with this, she also carried out an EDU post-chase experiment, where EDU is a thymidine analog and only incorporated when cells are dividing. So doing this EDU post-chase experiment allowed her to find that activating FGF signaling in the glia niche also impacted on the ability of neural stem cells to produce the neurons. So there's a significant fewer neurons that are produced by the neural stem cells upon glia FGF activation. So together, this first piece of data shows that glia FGF signaling really inhibits neural stem cell proliferation. Then she went on to ask whether this phenotype is really specific to FGF activation, or if you increase the glia size by other means, could you also produce this phenotype? And what she found is that it is really actually highly specific to FGF activation. And this is mainly due to the fact that only FGF activation and not insulin activation or EGFR activation can cause an increase in the size of the cortex glia cells that surrounds the neuroblast. So there are really many different subtypes of glia cells. Some of them form the blood-brain barrier, but the cortex glia cells here in green really surround the neural stem cells, are the immediate microenvironment that surrounds neural stem cells. So it is only by increasing this subset of cortex glia cells can you see an effect on neuroblast cell cycle progression. So next, she wanted to identify the mediators of glia FGF regulation, and she took a candidate approach. And this is because it is already known that some genes in the lipid metabolism pathway have been shown to influence the behavior of neural stem cells. So back in 2015, it was showing Alex Gould's lab that organelles called lipid droplets are localized to the glial cells here labeled in green. So Joanna went on to ask if genes involved in lipid metabolism could also be mediators of this glial neuroblast crosstalk. And perhaps not surprisingly, upon glial-FGF activation, she saw a significant increase in the number of lipid droplets. And you would think this is because you've got more glial cells, right? But when normalized to the number of glial cells, she still saw a significant increase in lipid droplets. And then this is confirmed by some qPCRs, where she found a significant increase in lipogenesis genes such as fatty acid synthase 1, and a decrease in lipolysis indicated by the increased transcription of a gene called LSD2, which is an antagonist of lipases such as Brahma lipase. So altogether, it suggests in the glial cells, glial-FGF activation really sort of skews towards lipid storage and prevents lipolysis. So how does glial niche lipid metabolism impact on neurostem cell proliferation? She then postulated this could be via lipid modified signaling molecules. And one of such molecules is that of a very well-known patterning gene called hedgehog, which is post-translationally modified by PAR mutilation at its N terminus and cholesterol modification at its C terminus. So Joanna then went on to ask if hedgehog is localized to the glial cells and could it be playing a role. And when she looked at hedgehog staining, she found this very striking phenotype where this hedgehog staining appeared to in a ring sort of shape. And when she co-stained with lipid droplets, she found that hedgehog is ligands are actually localized to the surface of these lipid droplets. So these two, hedgehog is closely associated with lipid droplet organelles. And this specific localization or the association of hedgehog with lipid droplets seems to be highly specific to the glial cells, but not in other cell types in the fly. So what this suggests is that hedgehog could be playing a role, and if it does so, then you would suggest it would suggest that hedgehog signaling must be active in the neurostem cells, and this appears to be true. So Joanna then went on to have a look at a readout of a hedgehog signaling called cubitus interruptus. And CI like this seems to be very much active in the neurostem cells, here co-localized with neuroblast marker called Miranda. And when she autonomously activated hedgehog signaling in the neuroblast, she also saw a slowing down of the neuroblast cell cycle progression. So together, this suggests that the phenotype we saw through glial-FGF signaling may be accounted for by downstream hedgehog signaling. So to test this, she thought it would be interesting to look at whether there's increased hedgehog in this context of glial-FGF activation. And indeed, there's a really dramatic increase in the amount of hedgehog in the glial niche. And in wild-type brains, when she overexpressed hedgehog in the glial niche, she also saw that neurostem cells really slow down its cell cycle progression. So altogether, this sort of tended to suggest that the phenotype we saw due to glial-FGF activation could be accounted for by downstream hedgehog signaling. So to directly test this, she utilized a nice genetic tool. This is a LexA-LexOp system. So in flies, there are actually three binary expression systems, LexA-LexOp, QUAS, and GAL4-UAS. And the utilization of a combination of these binary systems allows us to simultaneously conduct gene manipulation in different tissues at the same time in the same animal. So what she did here is using the GAL4US system, she knocked down hedgehog signaling with RNAis against CI. And here she saw the neural stem cells. So this is happening within the neural stem cells. And this caused an increase in cell cycle speed. In the adjacent compartment in the glia cells, she activated FGF signaling. And as we saw previously, this caused a significant decrease in the progression of neuroblast cell cycle. And if you put these two together, then by inhibiting hedgehog signaling in the neuroblast really overrides the effects of its niche, suggesting that hedgehog signaling lies downstream of FGF activation in the glia niche. So far what I've told you is that in the glia niche, you've got hedgehog ligands, and they're closely associated with lipid droplets. And upon glia FGF activation, you've got an increase of both of these. And activation of hedgehog signaling via increased hedgehog ligand causes neural stem cells to slow down its proliferation. So next, Joanna asked whether lipid metabolism also lies downstream of FGF signaling in the glia niche. So now these manipulations are all carried out only in the glia cells. And what she found is that, as we have shown previously, activation of FGF signaling in the glia niche slows down neuroblast proliferation. But if you knock down key enzymes in the lipid metabolism pathway, FASN, DGAT, and LSD2, then you can significantly rescue the effect of FGF activation on neuroblast cell cycle progression. So this suggests that lipid metabolism genes also lies downstream of FGF activation in the glia cells. So finally, she went back to test the hypothesis that it is post-translational modification of the hedgehog molecule that is critical for the ability of hedgehog that's made in the glia niche to activate hedgehog signaling in the neural stem cells. And here, what the first thing she did is she directly tested if you knock down hedgehog ligand in the glia niche in the background of FGF activation, could you see a rescue? And this seems to be true. So when she used RNAis against hedgehog, against the hedgehog ligands, she saw that by depleting the amount of hedgehog ligand, you could significantly rescue neuroblast cell cycle progression. And due to time limits, I also don't show, but she also sees that the readout of hedgehog signaling in the neural stem cells is also rescued by this assay. And next, she asked whether power mutilation of hedgehog is important for the ability of hedgehog to signal between the glia compartment and the neural stem cells. And here she utilized a reagent called RASP, which is an acyltransferase that is very important for power mutilation of hedgehog on its N-terminus. And so what she saw by RNAis against RASP, she saw that power mutilation is indeed important. And if you don't have a power mutilated form of hedgehog, then the hedgehog molecule cannot signal to the neural stem cells to affect its cell cycle speed. So you're really ablating, this really lies downstream of glia FGF activation. Finally, she wanted to really test directly whether power mutilation of hedgehog is important. And here she utilizes a reagent, which is GFP labeled hedgehog N. Hedgehog N is a power mutilatable, but not cholesterol modifiable form of hedgehog. So in this case, it still can be power mutilated, but cholesterol modification just doesn't happen. And what she found is that when she activated, she overexpressed hedgehog N alone in the glia niche, she saw a very similar effect to overexpression of the full-length hedgehog, i.e. that neural stem cells significantly slow down their rate of cell cycle progression, suggesting that this palmitolatable form of hedgehog N is important to administer the function of glia neuroblast crosstalk. And then the next thing she did is that she asked whether palm mutilation in this context is relevant. So upon overexpression of hedgehog N, she then knocked down RASP, which is the enzyme that palm mutilates hedgehog N. And in this case, she's able to significantly rescue the effect of hedgehog N on neuroblast proliferation. And finally, she did one more experiment, which is by knocking down fatty acid synthase. So palmitate is either dietary derived or it's made through fatty acid synthesis, through fatty acids, FASN1. And so in this case, she overexpressed hedgehog N, but she knocked down FASN to deplete the amount of palmitate that's capable of palm mutilation. And in this case, she again saw a significant rescue. So altogether, this suggests that palm mutilation of hedgehog is really important and instrumental for the ability of hedgehog to travel from the glia compartment to the neuroblast. And while we do not know some of the mechanisms downstream of this, one observation we have made in this context is that there are many fewer punctures that actually make contact between the glia cells that are secreting this hedgehog and the neural stem cells. So suggesting there are fewer hedgehog and that can make contact with neural stem cells upon RASP or FASN knockdown. So, so we think the main mechanism that mediates the ability of hedgehog signaling between the glia and neuroblast compartment is via palm mutilation of hedgehog molecule. And this happens mostly in the ER. In addition to this mechanism, we also have some preliminary data showing that at the step where palm-mutilated hedgehog is then assembled onto the lipid droplets, an enzyme called LSD2, which competes for spots on these lipid droplets against lipases, is also relevant. So if we overexpressed hedgehog, which, to remind you, causes neural stem cells to slow down. And in this context, if we knock down LSD2, we saw two things. First, the distribution of hedgehog really changed. And second, that this really was able to significantly rescue the cell cycle slowdown we saw caused by hedgehog overexpression. So we're still trying to understand how LSD2 modulates hedgehog localization on the lipid droplets. And we think the main mechanism could be that hedgehog and LSD2 definitely co-localize to the same spots on the lipid droplets. And so this could be a competition for space. And if LSD2 is there, then if LSD2 is overexpressed, then it maybe takes over the available spots for hedgehog to be tethered to the lipid droplets. So finally, this is our working model. And what we think happens is that in the glia cells, there are hedgehog molecules that are tethered to the lipid droplets. And upon FGF activation in the glia cells, you've got really increased number of both. And this causes a net increase in the overall lipogenesis and decrease in lipolysis. And this increase in lipid metabolism is responsible for modulating hedgehog ability to signal to neural stem cells by two mechanisms. The first is by post-translational modification by palm mutilation, and the second is perhaps through competition for space on the surface of lipid droplets by LSD2. And then this really affects the ability of hedgehog to travel from the glia compartment to the neural stem cells, and its ability to activate hedgehog signaling in the neuroblast. And upon activation of hedgehog signaling in the neuroblast, we've got the slowing down of neural stem cell proliferation. And so overall, what we think is happening is that the glia cells and the lipid droplets are probably doing something to really constrain the amount of hedgehog that is available to be secreted from the glia cells to activate to modulate the ability of neural stem cells to progress through the cell cycle. As we don't have much time, but we really also have some data showing that this amount of hedgehog in the glia cells is really intricately balanced. So as I've shown you here today, excessive hedgehog molecules is a problem because it really slows down neuroblast progression. But if you don't have any hedgehog, then the formation of the glia chamber is actually affected also. So really, you have to keep a really moderate amount of hedgehog, and this could be one way of modulating how glia cells can modulate neural stem cell proliferation. So I'd like to wrap up by thanking the people who did the work, and primarily Joanna, who really did maybe 90% of the work I talked about today, and supported by a number of other members of the lab. We'd like to thank the people who generously share their reagents, the core facility at Peter Mac, the stock centers, and Austros for stock quarantine, and finally, my funding bodies. And thank you very much.
Again, fantastic work, Louise. Thank you very much for that. We're running a little bit late on time, but if there's any questions for Louise, let's see.
I have a question.
Yep, fire away, Sean.
Hey, Louise, in one of your images, it looked like the lipid droplets were extracellular. Is that true, or are they always in the glia?
So they're basically in the cytoplasm of the glia cells, and they are either tethered to the lipid droplets or they are just free flowing. So yeah, it would be really nice to really have a look at what organelles. We think they are in vesicles for sure. I mean, that's obvious, and they do co-localize with all these different Rab proteins, but that's how they travel, I imagine. But yes, I think they are in the cytoplasm.
So are there enzymes that can remove the palmitilation from hedgehogs so that it could bind to patch once it gets, because you have this big patch of grease and you've got hedgehogs stuck to it, right? I don't know how it would be free flowing so that it could bind to its receptors once it gets outside the cell.
Yeah, I mean, I think at least we show palmitilation is really required for this ability of hedgehog to travel. We can see that upon palmitilation, hedgehog signaling within the neural stem cells is active. But yes, we do not know how that binding to patch is affected by palmitilation.
Right, Julian, do you want to ask your final question, mate?
Oh, it's the same as Sean, so I think that was addressed. Thank you very much. Yeah, Louise, I can still smell the grape juice agar. Well done.
Okay, and